Hello everyone, my name is Kiara Lewis, and today I will be discussing my project on utilizing OmpW to determine the presence of the foodborne pathogen Salmonella. This project focused on finding proteins that bind to the OmpW protein found in Salmonella. In the picture on the left is an E. coli plasmid containing OmpW, with the portion in red being the sequence used for this project. On the right is a yeast plasmid that contains possible binders. Using the rapids method, this project hoped to see an isolation of these binders so they can then be used for Salmonella detection. Before I go forward, I want to give a little information on why this project is important. Salmonella is a bacterium that can be found in foods that you eat every day, such as eggs, meats, and vegetables. Eating something contaminated with Salmonella can result in serious illness or possibly death. Salmonella is especially hazardous to pregnant women who can pass it to their child. And the main purpose of this project was to use OmpW, also known as outer membrane protein W, that is found in Salmonella to create a test that can then identify whether or not a sample contains the bacterium. And this project utilized the rapid affinity pair identification via a directed selection process, also known as RAPIDS. To first begin this project, we had to start with determining the protein we wanted to use, which you now know as OmpW. Then we purchased E. coli with the protein inside. We streaked the plate with the E. coli, allowed colonies to grow, picked the colony, and placed it in media that would express and grow our protein. Next was protein purification. We used a sonicator to lyse the cells, releasing our protein, and then going through a chemical process to ensure anything excess was gone. To make sure the protein was properly purified, we ran a gel to ensure we saw a band only where we expected. A BCA assay was also done to check the concentration. Then we started the rapids method. The first part of the rapids method is the magnetic bead sorting process. The way this process works is by coating beads in an antigen for 2.5 hours and then putting these beads inside yeast cells containing different binders. After the incubation time, the beads were removed from the yeast and expected to contain things that bind to our antigen. On the picture on the left, you can see the bead sorting process cycle. There are positive and negative bead sorts. Positive sorts are what was previously mentioned. Negative sorts are putting naked beads inside of the yeast to see what binds to the beads and then getting rid of it. In the table, you can see the calculations from the bead sorting process. From previous work, we knew we wanted 10 nanomoles of protein. The calculations were then done to show the volume of protein we desired. On this slide, you can see the results of our protein purification and magnetic bead sorting processes. In the top left corner is a picture of the gel that we ran to see if our protein was purified. So the thing on the furthest on the left is the ladder, which is just used as a reference to see the molecular weight of what we loaded. And on the right, you can see two bright bands. And since these bands are around 21 kD, we knew that this was our properly purified OmpW. In the graph labeled BCA as a standard curve, and the table under the picture of the gel is what helped us determine the concentration of this purified protein. You can see it was 32.3 mg per mil, and this is extremely exciting because this protein can be used for a long time. And finally, the table at the bottom right is our library diversity before and after the magnetic bead sorting. So you can see it went from 1.4 times 10 to the 9th to 3 times 10 to the 5, which shows that the bead sorting process worked properly. There are three main things I want you to take away from this presentation. One is that OmpW is a useful protein to use when trying to detect Salmonella in a sample. The second is that magnetic bead sorting is a great process to use when isolating binders to any antigen, though OmpW is our protein of choice here. And the third is a test for Salmonella isn't just useful for companies, but for their consumers as well. Some next steps include sequencing the yeast after the bead sort. So we know that we have binders, but we don't know what they are, and this will be helpful in determining what these binders are. Next would be to do fluorescence activated cell sorting, also known as FACS. So this helps us narrow down which binder is most efficient, which binder is binding the best. And third would be to do the agglutination assay, which is just a test. So on the right, you can see a picture of a before on the left and after on the right. So on the left is before the sample is introduced to the test. If you would shine a light through the sample, the light will pass straight through. It'll be clear. However, on the right is after the sample hasn't been introduced. So everything begins to clump together. And if you were to shine a light, things would be a little cloudy. This page just shows two of the references that were used to aid in this project. For their help, I would like to thank Dr. Hadley Sykes, Yenning Howe, Esteban Bermudez, and Christian Lubin. They played critical roles in the success of this project.